Hello, everybody. I'd like to thank Mona and Mark for inviting me to give this talk. I'll be talking about root microbiomes as drivers of Spartina alterniflora production. And I'd like to highlight my PhD student, Jose Rolando, who's done much of the work that I'll be presenting today. We study those microbes that are intimately associated with the very important salt marsh plant Spartina alterniflora, which is the dominant marsh plant on the coast of Georgia. And first, I want to remind everyone that healthy salt marshes cover much of the Georgia coast, about 400,000 acres. And this comprises about 1/3 of the healthy salt marsh on the east coast of the United States. I also want to remind everyone about the services provided by salt marshes to people, which include the fact that marshes are a very important nursery ground for fish and shellfish that we like to catch and eat. They also provide a very important physical barrier that protects coastal communities from catastrophic storms like hurricanes. That is, salt marshes act to disperse or dissipate the tidal energy of storm surge as it approaches the coastline. And lastly, I like to say that salt marshes are the kidneys of the coast, acting to remove pollutants from water as the water transits from rivers and streams across the coastal zone and into the coastal ocean, protecting water quality. So we want to understand plant microbe interactions and how microbes help plants to grow better. And in order to do that, we've been studying these interactions along a classic gradient of tall Spartina to short Spartina that exists in many coastlines. And so here you see the zonation in this cartoon with the tall form of Spartina growing next to a large tidal creek, and then the Spartina becoming stunted as you move inland in the marsh. It's well known that this gradient is linked to stressors of plants that include soil anoxia, sulfide, and salinity. So roots need oxygen to breathe, just as we need oxygen to breathe. And so where there's no oxygen, roots have to metabolize anaerobically, and that is less efficient, causing stunted plants. Sulfide is an important phytotoxin for plants, and also salinity acts as a stressor. All of these stressors are impacted by chemical exchange between soils and the overlying water in salt marshes. That is, you have more exchange along the large tidal creeks because of tidal flushing, as well as crab bioturbation. Crabs build burrows. Those holes aerate the soil, and the aeration helps the plants to grow better. So our objective in this project is to investigate the role of the microbiome in supporting the health and ecosystem function of Spartina. And we've been doing that at Sapelo Island and Skidaway Island, two barrier islands in Georgia. We set up, as you can see in this picture, transects from the tall to short Spartina on Sapelo and on Skidaway, and we've been studying these transects over the last several years. And you can see in the lower right-hand photo here how we expose the root zone and sample the root zone and study the microbes in the roots. Here I'm showing some results from this study. And first of all, look on the upper panels here. Notice that in all of these graphs, we plot the parameter versus the form, the growth form of Spartina from short to tall. And so you can see that there are much higher numbers of crab burrows along the large tidal creeks where the tall form of Spartina grows. We see higher snail density rather near the short form of Spartina, and we think this is another stressor impacting the health of the grasses. On the bottom panels, here you see pore water chemistry. So sediment EH is an indication of anoxia, and so you can see that the soils are most anoxic where the short form grows, and then that increases to the large tidal creeks where the tall form grows. Conversely, we see very high dissolved sulfide levels, this phytotoxin, where the short form grows, where the Spartina is stunted, and then those sulfide levels get much lower as you get where there's more tidal flushing where the tall Spartina grows. And also we see elevated salinity in the inland of the marsh where there's more ponding. Here we have measured rates of microbial activity using enzyme assays. So 3 different enzymes are plotted here versus plant biomass as a proxy for Spartina production. And in every case, you can see microbial activity directly correlates with production of the grasses. And that leads me to my first conclusion, which is that we see enhanced microbial activity in the rhizosphere, and we think that that replenishes nutrients as well as electron acceptors in the higher biomass stands. Next, I'd like to talk about how we use sequence-based methods or metagenomics to understand the microbial communities across these different growth forms and in different compartments of the plant. What metagenomics is basically we sequence all the genes for all the organisms that are present in a sample, and we can use these metagenomes to reconstruct the genomes of organisms that we can't culture, so we don't have to culture them to study them, and to understand their metabolic potential. We have collected over 200 billion base pairs of DNA sequences from the salt marsh, from the Georgia salt marsh. We've described over 160 species, many of which are new to science, have not been studied before, and we think this is a very important resource for studying marsh microbes. So first I'm showing here some results on the diversity and abundance of microbes associated with Spartina plants. And if you look in these upper 2 panels here, you can see that we've studied diversity and abundance not only according to the growth form of Spartina, but also according to compartment, from farther away from the roots in the bulk soils or sediments to the rhizosphere, which is the soils that it's attached to the root, to the endosphere, which is the root itself. And what you can see is both microbial diversity and the abundance of microbes determined with qPCR declines as you go into the endosphere. And we think that this is a gating effect whereby the plants actually select for certain microbes to intimately associate with inside the roots. You can also see here that we see higher microbial biomass in the tall form of Spartina in the bulk sediment in the rhizosphere, whereas in the endosphere, in the roots, you see there's a switching and actually a higher abundance in the short form of Spartina. So we think that this indicates that the short form concentrates microbes in the roots to help them deal with stress. Here we have calculated what's called beta diversity and a Bray-Curtis distance metric, and this simply looks at how similar or different microbial communities are amongst these Spartina plants. Each dot represents thousands of gene sequences of microbes in that particular sample, and so here you can see that the microbial communities are very different across different plant compartments, from the bulk sediment to the rhizosphere to the endosphere. And also we see different communities in the short Spartina in red, tall Spartina in blue, and whereas the medium is kind of a mixture between the two. I don't have a lot of time to elaborate on this, but I want you to know that sulfur bacteria, since salt marsh soils are largely anaerobic, sulfur bacteria are very important to metabolism in the salt marsh and the biogeochemistry, and that includes sulfate reduction, which is the microbial respiration of sulfate to sulfide and the concomitant oxidation of organic matter to CO2. And also that sulfide is a very important storage, represents a very important storage of chemical energy. And so microbes through sulfur oxidation can harness that energy and produce microbial biomass. So these graphs show the relative abundance of different types of bacteria in the root zone of Spartina. And if you look in the top 2 panels here, you can see that sulfur oxidizing bacteria are concentrated in the roots in the endosphere and show a high relative abundance. And also sulfate reducing bacteria are also concentrated in the endosphere. So we think that there is this very rapid cycling of sulfur in the root zone that's important to the metabolism of Spartina. And in particular, I want you to look at this one type of sulfur oxidizing bacterium that's known as thiodiazotropha, which is concentrated in the root zone. Thiodiazotropha is important because it's a group of endosymbionts, so symbiotic bacteria that live inside of plant or animal tissues. And they've been studied mainly in clams of subtidal marine environments, present, for example, in seagrass beds. So these sulfur oxidizers live in the tissues of the clams, and they fix carbon and they fix nitrogen. They make energy by sulfur oxidation, and then they pass that carbon and nitrogen on to the clam in this case. And we think the same thing is going on in Spartina plants, that sulfur oxidizers are fixing carbon and nitrogen and passing that to the plant. And we have some further evidence besides sequence-based evidence for that. Here we've measured rates of nitrogen fixation in the various plant compartments and in the various growth forms. And you can see in every case we get the highest rates of nitrogen fixation in the root zone or endosphere, shown in blue here. And also we see that the rates are higher in the short Spartina versus the tall Spartina. Again, agreeing with what we see, that this concentration of microbial biomass and especially sulfur oxidizing bacteria in the root zone of the short Spartina. Our second conclusion then is that sulfur oxidizing endosymbionts benefit the Spartina plants by detoxifying the root zone, removing sulfide, and also by fixing carbon and nitrogen that can be passed to the plants. Lastly, I just wanted to say that we are very committed to supporting the extension, education, and outreach of Sea Grant along with research. And so we have, in the course of this project, developed interactive activities. We call this March Madness, collaborating with marine extension interns down in Savannah on this project. We developed these activities for the Atlanta Science Festival. Because of COVID, we weren't able to do those in person, so we hope to do those in person soon. We have taught activities in collaboration with Marine Extension, again in collaboration with their summer camps. We have participated in the schoolyard program every year at the University of Georgia Marine Institute, teaching teachers about salt marshes, and we've also given a number of webinar talks in various venues to talk about the importance of salt marshes. Lastly, I want to say that I want to talk about how we can implement what we have learned from the Sea Grant research. And so what we would like to do is use what we have learned to better develop strategies for shoreline stabilization and for restoring coastal habitat in Georgia. And for this, we plan to partner with the Living Shorelines Working Group. Some of the things we can do are to identify and characterize microbes that might cause disease in Spartina, either in the natural marsh or when the plants are propagated for restoration, and thereby developing strategies for sourcing, better strategies for sourcing and propagation of plants to use them for restoration. Lastly, I just want to thank Ann Lindsay and her team at Marine Extension for collaborating with us on extension, outreach, and education activities, and Merrill Alber and Clark Alexander for helping us to develop these field sites, and of course, Georgia Sea Grant for funding.